Technical Veterinary Learning TVL channel presents today Viral Diseases of Equine. This lecture presented by Professor Ahmed Mamdouh Al-Sharif, Department of Veterinary Medicine, Faculty of Veterinary Medicine, Bani Suef University. As we did before, we will subscribe the most updated veterinary knowledge about infectious diseases. So, OIE listed equine diseases and infections in force for 2020 are contagious equine metritis, taurine, equine encephalomyelitis, equine infectious anemia, equine influenza, equine pyroplasmosis, infection with African horse sickness virus, infection with equidae herpes virus 1, infection with equine arthritis virus, infection with Burkholderia malii, glanders disease, Venezuelan equine encephalomyelitis. All the names highlighted with yellow color are for equine viral diseases. We will speak today about the viruses affecting respiratory system of equines. These are equine influenza, equine rhinopneumonia, equine viral arthritis, equine rhinovirus, and equine adenovirus. We will speak now about equine influenza viral infection. Because the importance of this disease, this literature will present in three continuously parts. The first part of equine influenza literature will include explanation of the definition of the disease, the epidemiology, including pathogenic risk factor with complete explanation of the antigenic drift of the virus and how to occur. Equine influenza viral infection is an acute, febrile, highly contagious viral disease of upper respiratory tract of equines and rarely fatal in endemic areas. Acute means occur suddenly with sharp increase in the temperature to be elevated up to 40 to 41 centigrade. Highly contagious means transmitted by direct and indirect ways. Located only in the upper respiratory tract, nose, nasopharyngeal area, trachea, bronchi, and bronchiole with upper lobes of lungs. In endemic areas, mortality reaches to 1% only, but in epidemics, mortality may reach to 25%. Equine influenza, as all diseases that infect animals and other creatures on this earth, began with the creation of the universe, but scientific reports started from the 7th century. Equine influenza viral infection characterized by the following. Number 1, fever, where the temperature reached to 40 to 41 centigrade. Number 2, dyspnea, difficult in respiration process, and then persistent hacking cough, which reached to 2 weeks and may be longer to 6 months of age. Number 3, rhinitis, with watery secretion that it changed into mucoid and mucopurulent discharges due to secondary bacterial infection, as well as presence of conjunctivitis, which reported in most of cases because of the connection between eyes and the nasopharyngeal area through lacrimal ducts. Equine influenza viral infection had a great economical importance. Economical importance of equine influenza viral infection coming from loss of several training days, especially in racing horses, polo gamers, show horses, and those used for military purposes. As the course of this disease may reach to 2 to 3 weeks and may extend up to 6 months of illness. Also, the economic cost of symptomatic treatment and vaccination of equine infectious disease, where the drugs used for equines are somewhat expensive. Mass vaccination every 6 months will cost both great veterinary efforts and a lot of money. Great epizootics of equine infectious disease was reported at 1872 with the pandemic of human flu that occurred at that time at the Earth. Public health risk of equine infectious disease are equine influenza viral infection showing a little risk to a few people coming in contact with infected horses developed the antibody to equine influenza virus. We will speaking now about epidemiology of equine influenza viral infection. Pathogenic risk factor of equine influenza. Equine influenza virus belongs to family Orthomyxoviridae, genus Orthomyxovirus, with 3 strains: A upon equine 1 H7N7, A upon equine 2 H3N8, and A upon equine upon Miami upon 1 upon 63. H7N7 strain of equine influenza firstly isolated at 1956, and since that time a very few cases were reported, though OIE recommendation by exclude addition of this strain to prepared vaccines against equine influenza. H3N8 strain and Miami 1 upon 63 strain firstly isolated and reported at 1963 in United States. H3N8 strain since isolated and reported at 1963 and up to 2019 caused huge outbreaks throughout the world, though OIE recommendation by making AI vaccine from clade 1 and clade 2 of this strain. Last recommendation and reported of OIE organization that equine influenza virus with strain H3N8 were isolated and or characterized from outbreaks in 2019 from the following countries: Argentina, Chile, and Uruguay from South America. In Asia, China was reported, but from Europe, France, Germany, Ireland, Netherlands, Sweden, and UK. Nigeria in Africa was reported, as well as United States of America. This is a diagrammatic structure of equine influenza virus. We will see lipid bilayer and then nucleocapsid protein with segmented stranded RNA gene, hemagglutinin, neuraminidase, and ion channel with matrix protein. This is a 3D diagrammatic structure of equine influenza virus. We will see lipid envelope, the capsid, RNA protein, neuraminidase or slidase with hemagglutinin and the ion channel. This is the structure of equine influenza virus under electron microscope after staining by gold. This is another picture of equine influenza virus. (3D structure of equine influenza virus). We will see here the neuraminidase and hemagglutinins with lipid envelope. This picture clarifies the presence of equine influenza virus in the bloodstream of infected equine. Every horse said to his partner, "Oh, skip barrier is very hard this year because of presence of horse flu outbreak." Antigenic drifts of equine influenza virus leads to continuing susceptibility of equines to infection with new strains and substrains, as well as it causing reduced efficacy of prepared vaccines, even with OIE recommendation, and this is proved through multiple outbreaks that occurred during 2019, 2018, and this year, 2020. In this picture, the process of antigenic drift of equine influenza virus are clarified. Binding of the viral particle by hemagglutinin and neuraminidase to the host cell, either a specific or non-specific host. Then host cell engulf the viruses by endocytosis process, and then uncoating and diffusion of the virus with cellular host content will be occur. Messenger RNA of the equine influenza virus control the host genome and began to produce new numbers of equine influenza viruses. Entrance of several hosts by these viruses for certain period of time will lead to occurrence of antigenic drift and formation of new strains of equine influenza viruses, and this is the cause of several outbreaks occurred during the last decade. This picture explains the antigenic drift process by infection of specific and non-specific hosts by equine influenza viruses. Equine influenza viruses survive in low temperature and humid areas. Equine influenza viruses remain alive in aerosols coming by sneezing of infected equines for about 24 to 36 hours. Equine influenza viruses are sensitive to heat, so increase of temperature will reduce the multiplication and the transmission of them. Also, lipid solvent and formalin could be used in hygienic disinfectant processes of equine harness. Dear veterinarians, dear student of veterinary medicine, second part of equine influenza will be continued. Please subscribe of this channel and listen to the whole lecture. Thank you and see you later.